going to talk about some major organ systems in craniates or invertebrates, but to do that, we really need to talk about why at least some of these major organ systems need to be there at all. In single-celled organisms, you don't really have much in the way of organ systems. You do a little bit. The organelles in some ways are functioning in a similar way to major organ systems, but it's really on a very different scale. And what it tends to come down to is surface to volume ratio. So the surface to volume ratio is really a property of geometry. It's not a property of biology, it's a property of geometry. And because of surface to volume ratio, because of what it means to something as it gets larger, you sort of have to have certain organ systems evolve to deal with those physical problems that surface to volume ratio imposes on living things. So if you look at the across the top of this table, you've got some different cubes. The shape doesn't matter. Cubes are usually used just because cubes, it's easy to do math with a cube. That's the only reason. The same relationships would be the case with any other shape, as long as the shape doesn't change. The only thing that's changing here is the size of the cube. That's the important thing. So you can imagine that each cube is like a cell. Okay, so here's a kind of a small cell, here's a much bigger cell, here's something made out of a whole bunch of cells. So you can think of them that way. Now the units also don't really matter. You have the small cell is 1 unit on a side. It doesn't matter what the units are. You could think of it as millimeters or centimeters, whatever you want. The larger cube or cell is 5 units on a side, and there you go. Now let's start with the small one. So let's take a look at this one. If that's a cell, there are materials that have to get into the cell, and all those materials have to pass through the surface of the cell. So they have to, which is going through the cell membrane, but that's the surface area of the cell. And those resources are being used by what's going on in the volume on the inside of the cell. So it's easy to imagine just what's going on with oxygen. The cell has a metabolism that's using oxygen, so all the oxygen that the cell uses has to pass through the surface area to get into the cell from the outside. And in the process of that metabolism, one of the waste products is carbon dioxide, which is toxic. If it builds up too much, it's toxic. So that has to get out of the cell. Now, to do that, that carbon dioxide also has to pass through the surface area of the cell. Now, the other half of that equation is how much oxygen does the cell need and how much CO2 does it need to get rid of? That's dependent on the volume of the cell. The bigger the volume, the more oxygen it needs and the more CO2 it needs to get rid of. The bigger the surface area, the faster those materials can pass either in or out of the cell. So let's calculate the surface area of this cell. Since it's a cube, it's really simple. So the surface area is just the surface area of all the facets of the cube. So if the cube is 1 unit on a side, 1 times 1 gives you, let's just call them centimeters just to make it easy. So 1 square centimeter of surface area, but that's for only one face of the cube. And the cube actually has 6 faces. You can see 3 of them, and then there's 1 at the bottom and then 2 behind. So it has 6 faces total. So 1 square centimeter of surface area for a face times 6 faces gives you 6 square centimeters of surface area. Now, that is a measure of how fast materials can pass in or out of the cell. If you look at the volume of the cell, now, remember the volume of the cell is a measure of how fast the cell needs to get, or how fast the cell is using oxygen and how fast it needs to get rid of the CO2 that it's making. So to figure out the volume, it's 1 times 1 times 1. It's 1 cubed for the volume, which is pretty easy to do the math on that number. It's just 1 cubic centimeter of volume, since we're just calling them centimeters for units. So there it is down there. There's our volume. Now there's a relationship that's important because the speed that materials can enter or exit the cell is determined by the amount of surface area. The rate that the cell either needs those materials coming in or needs to get rid of the waste materials is determined by the volume. So what you do is take the surface to volume ratio, which is just taking the surface area, which is 6, and dividing it by the volume, which in this case is 1. So 6 divided by 1 is 6. There you go. There's the surface to volume ratio for that small cube. Now let's say that there's selection pressure to make this small single-celled organism into a bigger organism. So one way you could do that would be to just take that single cell and just allow it to grow bigger and bigger and bigger, right? Just keep everything pretty much the same and just make it bigger. And that's what we've done in the middle column here. You've got the same cell. The only difference is it's larger. Each side is 5 times longer than in the small cell. So in that big cell, if you calculate the surface area, it's 5 times 5 is 25 square centimeters, but that's only for one face of the cube. And of course, it also has 6 faces, so 25 times 6 gives you 150 square centimeters of surface area, which is not too surprising because it's bigger, it has more surface area. Not very surprising. Now, let's do the volume. To calculate the volume, what we have to do is just take the length of a side cubed, so 5 cubed. 5 times 5 times 5 is 125. So again, it's got more volume than the small cube. Not very surprising because it's bigger. So there you go. It's bigger, so it has more surface area and more volume. However, when an object gets bigger, the relative amount of surface area the surface area is increasing, but the surface area is not increasing as fast as the volume as the object gets bigger. So if we figure out the surface to volume ratio now, take 150 and divide it by 125, now it's only 1.2 if you do the math. So in other words, the amount of surface area per unit volume has gone down, and it's gone down significantly. And again, remember, that's a problem because as the volume is increasing very, very rapidly, that means the oxygen demands are increasing rapidly, and the amount of carbon dioxide waste that the cell is making is increasing dramatically. So we have to move oxygen in and CO2 out faster than we did before to keep up with the demands and the production of those molecules. And the problem is that the surface area is getting bigger, but it's not the increase is not keeping up with the increase in volume. And so very quickly you get to the point where you are not able to move materials in and out of the cell fast enough to keep up with the demand, and the cell is going to die. If you've seen paramecium, which I'm sure you have, you can imagine paramecium, to me, they're big single cells. For single cells, they're very large. But if you look at them, they're almost whale-shaped. If you wanted to make a whale, well, you could start with a paramecium, and you're not too far from a whale with the paramecium. You really just have to make it a lot bigger. But if you try to keep it one cell, it's not going to work. So there's a way around the surface to volume ratio problem, and that is, well, to basically break the rules. The rule has been that we're making the object bigger, but we're not changing the shape. Well, what if we change the shape? So why is a whale bigger than you, or bigger than a paramecium? It's because it's made out of a lot more cells. The cells are not any bigger. Well, and remember, a paramecium is kind of a weird cell, but if you compare, a cheek cell from a whale with one of your cheek cells, if you looked at them both on a microscope slide, you wouldn't really be able to tell the difference. They're going to be the same size and everything. If you pulled a kidney cell out of yourself and a kidney cell out of a whale and put them on microscope slides and looked at them, they're going to basically look the same. The difference between you and a whale is mostly that the whale has a lot more cells. The cells are not bigger than your cells. There's just a lot more of them. So take a look at the last structure here. This is not a single cube anymore. This is a cube that overall is the same size as the large cube, but it's made out of little cubes, and each of those little cubes is the same size as the original small cube that we were looking at. So if you want to figure out the volume of this, it's the whole structure is also 5 units on a side, just like the middle cube was. So the volume, you'd calculate it the same way. 5 cubed is still going to be, it's going to be the same. 125, it's going to be the same as it was for the middle cube. But when you figure out the surface area, because these cubes are separated a little bit, all the little cubes are separated a little bit, when you figure out the surface area, you've got to figure out the surface area of each little cube and multiply that by the total number of cubes that are in this whole structure. And if you do that, the surface area is actually much higher than it was in the middle cube. And if you do the math now, so our surface area is 750 square centimeters. Our volume is 125 cubic centimeters. If you just divide 750 by 125, our surface to volume ratio is 6. It's the same as it was for the little cube, even though it's a much larger structure. The structure itself is as large as the big cube. Now, that only works if you have organ systems that are going to bring materials to all of these cells, and each of these little cells in this structure can in some ways act like a separate individual cell, like the original one that we had here. And that works because, well, certainly in vertebrates you usually have a blood supply, so your body does not have enough surface area compared to the volume of your body for oxygen to just diffuse in through your skin. Your skin is actually way too thick, and it's not very permeable to oxygen anyway, but even if it was, uh, you're just too big. You don't have enough surface area compared to the volume for that to work. So what you have instead is you've got a blood supply that is circulating blood basically up to almost every cell in your body. There's almost no place in your body where you have uh, a cell that's more than about 2 cell diameters away from your circulatory system. And you've got to keep the oxygen levels high in the circulatory system, and you've got to keep the CO2 levels low. Well, how do you do that? Well, you've got lungs, so that blood is circulating through the lungs, and your lungs are basically, it's a bunch of surface area that's folded up and very finely divided inside your body, so that it massively increases the amount of surface area. You have way more surface area in your lungs than you have just out in your skin on your body surface. So this is a way around the surface to volume ratio problem. If you look at this as a function, so this is just using cubes. So here's the small cube that we had with the length of a side was 1, and our surface to volume ratio is 6 for that one. And then if we go to the larger cube where the length of a side was 5, and here we are at 1.2, our surface to volume ratio. But you notice that as that object keeps getting bigger, the surface to volume ratio drops dramatically. I mean, look at this here. What's the surface to volume ratio there? I mean, it's what, maybe 0.3 or 0.4, something like that. And that's only 15 times bigger than a single cell, than a normal size cell. How many times bigger than a single cell is a whale? Many hundreds of trillions of times bigger than a single cell. So the surface to volume ratio of a whale, it's not quite 0, but it's so tiny, it's basically asymptotically approaching 0. It's approaching 0. It's getting very close to 0. Surface to volume ratio um, also relates to strength, and so one of the applications of surface to volume ratio is what they call strength to mass ratio. So you're looking here at two front leg skeletons of 2 different mammals, and all mammals, and basically all tetrapods, have pretty much the same skeleton. It's modified a little bit. Sometimes some bones are separate in one species and they're fused together in another, but we all have pretty much the same basic skeleton. It's just been modified a little bit. So if you look, this is the scapula or the shoulder blade in each of these animals. This is the humerus. Then you've got the radius and the ulna down here in the, well, what in us would be the forearm. In this animal, those 2 bones are there, but they fuse together. And then you've got the bones of the wrist here, and then the toe bones down here. So you've got the same bones. Now, in this picture, these 2 drawings are scaled so they're the same height. But you can tell when you look at it which one is from a bigger animal, which one is the bigger mammal. So look at it now and decide whether it's the left one or the right one that's from a bigger mammal. The one on the left is the front leg of an Indian elephant. The one on the right is the front leg of a deer mouse. Now, here's the thing. All these animals are built on the same basic plan. An elephant and a mouse have the same basic skeleton. They've got the same basic tissues. Because they're both mammals. They're pretty closely related. Um, but you can tell the difference even when the bones are scaled to be the same length. You can tell which one's from a bigger animal. Now, think about the elephant. Why is the elephant bigger? 1 cubic centimeter of elephant tissue is going to weigh the same as 1 cubic centimeter of mouse tissue. Why is it that the elephant is heavier? Because it has more volume, more cubic centimeters of tissue. It's not that each cubic centimeter is heavier, there's just a lot more of it there. So the mass of the elephant is dependent on the volume. And so is the mass of the mouse. It's dependent on the volume of the animal. An elephant has a lot more volume, so it weighs a lot more. Now, the skeleton has to be able to support all of that weight. Now, the strength of these bones has really nothing to do with the length of the bones. That's not where the strength is. Imagine cutting directly across through this bone and looking at the surface area of that cut edge. Or cutting through this one and looking at the surface area of that cut edge. That surface area is telling you how strong the bones are. If it's thicker, if it's got more surface area when you do the cross section, then it's a lot stronger. And so imagine doing an experiment here where you take a mouse, a deer mouse, and put a deer mouse on the table and let go of it, and it's going to jump off the table, and when it hits the ground, it's going to keep right on running. It's not even going to sprain its ankle. And if you think about it, like a normal table height is probably 8 or 10 times the body length of a mouse, not counting the tail, just the head and body. Now take an elephant and chase it off a cliff that's 8 or 10 times the length of the elephant high. And of course, the elephant is not going to keep running when it hits the ground. It's just going to be a big pile of crushed bones and smashed tissues. And that's because of the surface to volume ratio. A mouse is, in an absolute sense, it's got a much weaker skeleton than an elephant does. But relative to its body mass, the mouse's skeleton, even though these bones that you see here are so thin, the mouse's skeleton is actually proportionately much stronger than the bones of an elephant. Elephants have thicker bones. They are absolutely stronger, but elephants are actually, their skeleton is kind of on the edge of how strong it needs to be, because it's so metabolically expensive to keep making these bigger bones. You need so many minerals to make them, and they're so heavy to carry around all the time.